Welcome to video 4, where we will focus on cell structure, and that is 1.2 of your unit guide. Okay, so 1.2 is looking at the ultrastructure of cells, and in that we're going to see what different types of cells are composed of, looking at individual organelle and things along those lines. So this is unit 1.2. So there you'll see your understandings, applications and skills, guidance, and all the other sections that we'll be covering. So first up, we're going to look at types of cells. Okay, now there's two main different types of cells, okay? They're either what we call eukaryotic or prokaryotic. Now, the prokaryotics that you'll see on the left-hand side there, they lack a nucleus and internal organelles, whereas our eukaryotic cells contain a membrane-bound nucleus and they also have membrane-bound organelles, so they're somewhat more complex. If we look closer at our prokaryotes, basically all bacterial cells are prokaryotic. Prokaryote DNA is located within the cytoplasm of the cell, and it's just one long chromosome. So it's a twisted circular chromosome. When this replicates, it will then separate and divide into two new cells, and that's what we call binary fission. Now, binary fission is prokaryotic reproduction, basically. Okay, so how our prokaryotic cell reproduces, the DNA in the circular chromosome replicates, and then that prokaryotic cell divides into two with DNA in both new cells. That is known as binary fission. So if we look at the structure of our prokaryotic cells, they have a cell wall. Okay, and this surrounds and protects the cell from bursting. They have a plasma membrane which acts as that barrier, allowing the movement of materials in and out of the cell. They've got a cytoplasm where all those metabolic reactions occur, but also remember within the prokaryotic cell, that's where the genetic material, the DNA, is found. Now, the prokaryotic cell has a nucleoid, and that is a region of the cytoplasm that contains that naked DNA as such. Okay, bacteria also contain small circles of DNA called plasmids. Now, they can pass from cell to cell. So you'll see there in the diagram, you'll see a plasmid identified, and you can also see where that nucleoid is. Now, prokaryotic cells also have 70S ribosomes, okay, and it's here that they synthesize proteins. Our prokaryotic cell also has the flagellum. Okay, these are projections out of the cell wall that enable the cell to move. Our cell also has what we call pili. Now, these are also projections out of the cell wall, and it allows the cells to connect together, and it also allows the exchange of materials. So the flagellum is the longer projections which allow movement, and the pili are shorter, slightly shorter projections which allow cells to bind together and allows the exchange of material. Okay, so if we look at our eukaryotic cells, these have a much more compartmentalized cell structure when compared to prokaryotic cells. And the partitions, or what we call compartments, are formed by a single or double membranes. Now, there's advantages to being compartmentalized. Basically, certain substances required for different reactions can be concentrated into one certain area in the cytoplasm. Substances that could cause damage to the cell can be kept inside a membrane of an organelle. For example, lysosomes they contain these digestive enzymes. Now, if they got out, they could actually break down parts of the actual cell. So it's good that they can be compartmentalized. Also, pH can be maintained within a membrane, given the ideal conditions for certain chemical reactions to occur. Organelles with their contents can be moved around within the cell if they're enclosed in that membrane then, so they can move around in their own little environment. Okay, what organelles is it really important for you to know at this point? There will be more that you're introduced to as we move through our course, but at this point, the important organelles for you to know are compartmentalized regions of the cell that carry specialized functions. So these are our organelles, okay? So we've got our nucleus, we've got our rough endoplasmic reticulum, our rough ER, we've got our Golgi apparatus, we've got our lysosomes, we've got our mitochondria, our free ribosomes, our chloroplasts, vacuoles, vesicles, cilia, flagella, centrioles, and microtubules. Okay, let's find out a bit more about each of these. Okay, so first of all, the nucleus, and here we've got a diagram of a nucleus. We'll see it's got that double nuclear membrane, nuclear pores, chromatin within the nucleus, and that dense chromatin there attached to the outside. And you can see a micrograph photo of our nucleus there. Now, the role of the nucleus, you would have heard in the past things like it's the brain of the cell. Now, that's really not biologically correct, okay? The nucleus does control the cell to a certain extent, but it's where the genetic material of the cell is found. Now, our nucleus has this nuclear membrane. It's a double membrane containing these pores. Our nucleus contains chromosomes, DNA, histone proteins, uncoiled as chromatin, and densely stained around the edge of the nucleus. So you can see that chromatin really dense around the edge of the nucleus. The dark staining in the middle is what we call the nucleolus. Okay, so whilst looking at that micrograph photo there, you can see that dense material in the center, which is the nucleolus. The nucleus is where the DNA is replicated and transcribed into what we call mRNA. Here we have our rough endoplasmic reticulum. We can see that this is made up of ribosomes and cisterna. Okay, and we can see a picture of it there as well as the diagram. So we call rough endoplasmic reticulum rough ER or RER. Okay, and this consists of flattened membrane sac called the cisternae with 80 S ribosomes attached. The function of the rough ER is to synthesize proteins, a secretion out into the cell. So synthesize means produce, to make proteins. Proteins pass into the cisternae and are carried in vesicles to what we call the Golgi apparatus. So, seems simple that we should move straight on to that Golgi apparatus. Here we've got the Golgi apparatus, and we can see that it's made up of vesicles and more cisterna. Okay, sometimes the Golgi apparatus is called the Golgi body or the Golgi complex. You may hear those terms at any time interchanged. They mean the same thing. Now, the Golgi apparatus consists of flattened membrane sacs called cisternae with many vesicles nearby. The function is to process the proteins that are brought in from the vesicles from the rough ER. These proteins are then packaged into vesicles and carried to the plasma membranes for secretion. So it's kind of, imagine it as a production line. Okay, so the proteins are made back in our rough ER. Okay, so our proteins are made here, and then they're taken to the Golgi apparatus for distribution. Okay, next up we've got a lysosome. Okay, and you'll see in our lysosome we've got these digestive enzymes which are surrounded by a lysosome membrane. Okay, and remember, it's those digestive enzymes that if they wasn't surrounded by that membrane, they would actually break down parts of the cell. So our lysosome is a vesicle containing, so vesicle means a unit, okay, just a part of the cell, just a area enclosed in a membrane. So a vesicle containing digestive enzymes which are used to break down ingested food, break down organelles in the cell or even the whole cell once they're no longer productive. From our lysosomes we move on to our mitochondria. I have a little joke here. The mitochondria have got the power, okay? You'll realise why I said that in a moment. So the mitochondria, we have an outer membrane, an inner membrane, we have these crista projections in there, and our matrix in between the crista projections, and a great micrograph picture there. Okay, so our mitochondria has got a double membrane around it. Remember the outer and the inner membrane. The inner membrane forms the invaginations, which we call cristae, and fluid inside the mitochondria, so around the cristae, is what we call the matrix. The function is to produce ATP for the cell by aerobic respiration. These are the energy house of your cell, of any cells. Here we've got our ribosomes, okay, so some free ribosomes, and you can see the micrograph picture of them there. Okay, so they're called free ribosomes when they're not attached to any other organelle. If drawing them within a cell, just draw as dots to get the scale correct, so they're just small dots. ATS are the same as attached ribosomes, okay, so they're ribosomes but they're attached, whereas these are free ribosomes. Okay, they're not enclosed by a membrane nucleus, and ribosomes are constructed in the region of the nucleus contained within the nucleola. The function is to synthesize proteins for use within the cell to the rough ER. So again, they're used in that protein synthesis. Alrighty, my favourites now, we're on to some plant cells. Okay, and here we've got our chloroplast. You'll see some of our organelles in our chloroplast. We've got starch grains, stroma, we've got a double membrane again, and we've got our thylakoid. So, chloroplast has that double membrane. Contains these thylakoids with a flattened sacs of membranes forming sacs of what we call granules. Okay, fluid, there's a fluid filled space which is called our stroma. Now the function of our chloroplast is the production of glucose through the process of photosynthesis. Remembering that chloroplasts are only found in plant cells, okay, and only in photosynthetic plant cells. They're what gives plant cells their green colour, okay, due to the chlorophyll within the stroma. Starch grains can be seen after the cell has been photosynthesizing. So if the cells haven't been photosynthesizing, if they've been in darkness, okay, those starch cells won't be present. They only occur due to the production of glucose and then that transfer of that glucose to starch within the cell. Again, we are looking at some storage organelles here, and we've got vacuoles and vesicles. Okay, so we've got a large vacuole, we've got some small vesicles, and we've got a vacuole that's containing food there. Okay, so these are surrounded by a single membrane which then encloses some fluid. Large vacuoles can be found in many plant cells. Okay, a vacuole is generally a key component to plant cells, except some specialised plant cells. Some animals absorb food and digest in vacuoles, and some unicellular organisms contain vacuoles to expel excess water called contractile vacuoles. Vesicles are very small and are used to transport material once inside the cell. Okay, so you'll get materials from the cell membrane to where it's needed within the cells. They're like transport items. Okay, microtubules and centrioles. We've got two little pictures there. Microtubules are fibres that form the cellular matrix, holding organelles into position and playing a role in the movement of chromosomes. So the organelles within the cell need to all be held within position, otherwise with gravity and things like that, they'll just all squash down the bottom of the cell. So they do need to be held into set positions, and it's our microtubules that do this. Cells have structures called centrioles, from which spindle fibres form during cell division, and we'll look at that at a later date. Okay, looking at cilia and flagella now. Now these are whip-like structures projecting from the cell surface. Flagella are larger, and usually there's only one of them. For example, the towel on a sperm cell. Cilia are smaller, and there are quite often many present, so like small hairs on the cell. Both can be used for locomotion. Cilia can be used to create a current in the fluid next to the cell, because there's so many of them, as they wave backwards and forwards, they can make a current around the cell. Okay, so how do we know all this about cells? I mean, we've already said that cells are very, very small. So how do we know all this information about them? Well, our everyday light microscopes cannot magnify enough to see our prokaryotic cells. So to see these, we need to use what we call an electron microscope. Electron microscopes have a much, much higher resolution than our everyday light microscopes. Resolution is the ability to distinguish separate parts from things we view. Okay, so if we magnify a coloured picture, we can distinguish between those individual colour pixels making up the whole picture. And the same goes for when we look at tiny, tiny organelles under an electron microscope. We can actually distinguish between the different organelles within the cell. Okay, now when we are talking resolution in our microscopes, this image here shows us something that's good resolution, so we can actually clearly see the images, compared to a poor resolution. Now the maximum resolution for light microscopes is 0.2 micrometers, whereas the resolution of an electron micrometer is 0.001 micrometer. So you can understand how much clearer you would see through that. And if we look here at these pictures, we've got a micrograph. That's a photograph taken by a microscope, so that's a key point of what a micrograph is, and I have been mentioning them as we've been going through this video. So we've got a prokaryotic electron micrograph here, okay, and it's of an E. coli cell, and you'll see that's 1 to 2 micrometers in length, but you look at how clearly you can now see that, to the extent that you could then use that picture to draw a diagram of the cell itself, and that is how you'd be expected to draw that diagram from what you're viewing above. Once you've drawn the diagram, you would then identify the parts that you are looking at. Okay, so drawings of prokaryotic cells should show the cell wall, the pili, the flagella, plasma membrane, the cytoplasm, the 70S ribosomes, the nucleoid with naked DNA, and this, guys, is one of your skills as part of your IB course, the drawing of the ultrastructure of prokaryotic cells based on electron micrographs. Alrighty, so if we look at Unit 1.2, we can now see what we have done. We have got some skills which we are going to practice in the lab as well to complete this unit. Thank you for watching.